Welcome to the Deep Dive, where we pull back the curtain on some of life's most profound questions. Today we're tackling one that feels incredibly personal, yet it's built into the very fabric of our biology. Why do we age? Why does aging even exist? For most of us, the immediate thought is wear and tear. Things just break down over time. And while that's part of the picture, it's far from the whole story. What we're about to explore is a deeply embedded evolutionary narrative, a fundamental why behind our decline that's more intricate and surprising than you might imagine. Our mission on this deep dive is to truly understand the evolutionary why of aging. We're not just describing the process, we're digging into the ancient logic that shaped it. Specifically, we'll unpack three foundational evolutionary explanations: Sir Peter Medawar's mutation accumulation theory, George C. Williams's antagonistic pleiotropy, and Thomas Kirkwood's disposable soma hypothesis. This isn't a how-to guide for eternal youth, but rather an explanatory map designed to help you understand why natural selection has, in a sense, allowed aging to persist. And trust me, grasping these evolutionary roots is absolutely crucial because it illuminates why modern interventions aimed at slowing aging work, or why they sometimes run into unexpected roadblocks. It gives you a framework for thinking about future scientific breakthroughs. So how are we going to navigate this fascinating, sometimes counterintuitive landscape? We're going to kick things off with a compelling natural experiment involving some truly surprising creatures, opossums. This real-world case study perfectly sets the stage for the core evolutionary theories. From there, we'll unpack those foundational ideas, delve into the precise biological mechanisms that underpin them, and then explore how cutting-edge modern interventions attempt to cheat these ancient evolutionary trade-offs. We'll even journey into the incredible diversity of the animal kingdom to see species that stretch the very boundaries of longevity. And finally, we'll confront the critical trade-offs involved in pushing the limits of regeneration, especially that unavoidable tension with cancer risk. It's going to be an intellectual adventure, connecting history, biology, and even some mathematical principles that dictate life itself.
What's truly fascinating here is that we're moving beyond just describing aging, beyond simply saying cells accumulate damage, to understanding its fundamental evolutionary roots. It's about unpacking the logic that underpins why our biology seems to tolerate decline, why it actively prioritizes certain functions over long-term perfect maintenance. To grasp this, we need to understand the pressures and incentives natural selection faces. This isn't just about molecular pathways, important as they are, it's about the very calculus of survival and reproduction. And for those of you who appreciate the elegant, sometimes brutally logical systems in nature, this framework is incredibly satisfying. To really anchor this life history logic, we're going to begin with what I consider a perfect natural experiment: the Sapelo Island opossum study. This case beautifully illustrates a core principle: how external mortality risks shape the evolutionary investment in longevity. It's a real-world demonstration that connects directly to the theoretical principles we'll explore.
You mentioned the Sapelo Island opossums as a perfect anchor for this whole idea. What makes their story so compelling, and how does it illustrate this evolutionary logic right off the bat?
It's a classic for a reason. Imagine an island off the coast of Georgia, Sapelo Island. Here, populations of the Virginia opossum, the same kind you might see waddling through your backyard on the mainland, historically faced significantly fewer predators. Think fewer coyotes, fewer large birds of prey, less of that immediate extrinsic mortality pressure. This was the basis of some truly elegant fieldwork conducted by Stephen Ostad, published back in 1993. His experimental setup was deceptively simple. He did a direct field comparison between these island opossum populations and their counterparts on the mainland, where predators were much more abundant. And his core finding was profoundly insightful. He reported that the reduced extrinsic mortality, that is, fewer deaths from external causes like predation or accidents on Sapelo Island, directly correlated with slower senescence or slower aging in that island population.
Slower aging. How did he even measure that in opossums in the wild?
Great question. He observed several specific, quantifiable endpoints that clearly demonstrated this slower aging. The island opossums showed later declines in their reproductive performance, meaning they continued to reproduce effectively for longer into their lives. They also exhibited later declines in connective tissue aging. Ostad actually measured this biomechanically from their collagen, indicating their physical bodies were indeed aging at a slower pace. And critically, they showed significantly longer overall survival compared to their mainland relatives. This wasn't just anecdotal. The study rigorously spanned dozens of individuals per cohort, providing robust statistical power to the findings.
Wow, okay, so real physical differences, not just living a bit longer.
Exactly. And these findings were entirely consistent with what we call life history theory. The fundamental principle at play is that when the environment stops killing you young, when the immediate threat of predation or external hazards is significantly reduced, natural selection can afford to invest more in maintenance and future reproduction. There's a reduced urgency to reproduce quickly and die because the organism now has a higher probability of surviving longer. Ostad, notably, reported no conflicts of interest, and the study was a longitudinal field comparison, meaning he followed the same animals over time, which adds greatly to its integrity.
So if I'm thinking about this as a fundamental principle, what does this opossum study really tell us, the listeners, about the broader evolutionary why of aging? It seems to set up a really clear, almost testable question.
It absolutely does. This study sets up a powerful, falsifiable question for all of biology. If extrinsic hazards fall, does selection for maintenance rise, and does aging slow? This deep dive will explain why the answer is generally a resounding yes, how it arises from the basic mathematics of natural selection, and crucially, where biological complexity, like the trade-offs within our own bodies, complicates the seemingly clean story, preventing us from simply living forever just by avoiding predators or accidents.
And it also makes you wonder then, how does nature even decide how much to invest in longevity? It seems like a constant push and pull.
Precisely. This opossum example is a beautiful illustration of selection at work, demonstrating a clear, fundamental trade-off. When extrinsic mortality is high, when the odds of being eaten or dying from an accident are significant, the optimal strategy for an organism is to put its limited resources into rapid growth and reproduction. You pass on those genes quickly before something gets you, but when that pressure is lifted, when death from external causes becomes less likely, selection can then favor diverting resources towards somatic maintenance, essentially, keeping the body in better repair for a longer time because there's a higher chance of future reproduction. It's a dynamic allocation of biological capital, driven by the environmental pressures an organism faces.
This next idea really shifts our perspective, and for me, it's where the deeper evolutionary logic truly becomes clear. If selection weakens with age, what does that mean for our biology? It just feels inherently counterintuitive. You think about evolution always pushing for perfection.
It is counterintuitive if you think perfection is the goal. But the core idea behind the evolutionary perspective of aging is remarkably simple. Natural selection's grip weakens dramatically with age. It's not about making us perfect or immortal. It's about making us good enough to reproduce and pass on our genes to the next generation. Once that crucial task is largely complete, the selective pressure to maintain a perfectly functioning body simply isn't as strong.
Okay, so who first really pinned this down?
This concept was formally articulated by the brilliant W.D. Hamilton back in 1966. Hamilton formalized what he called the force of selection on survival and reproduction. His core principle states that this force, this selective pressure, declines precipitously after sexual maturation. The reasoning here is based on straightforward demographic logic. As an organism ages, fewer and fewer individuals remain alive to enjoy any potential late-life benefits, such as continued reproduction or extended survival. Therefore, any genetic traits, whether they're beneficial or harmful, whose effects are delayed until late in life, contribute significantly less to an organism's overall fitness. Evolution simply has less interest, you could say, in what happens to you after you've passed on your genes.
So bad things happening late don't get weeded out as effectively.
Exactly. The consequence of this declining force of selection is absolutely crucial for understanding aging. Harmful alleles, or gene variants, whose negative effects only manifest late in life, are only weakly opposed by selection. They can persist and even accumulate in a population because their detrimental effects occur after most individuals have already reproduced. In stark contrast, early life benefits, even if they carry significant late life costs, are strongly favored by selection because they directly enhance an individual's ability to survive and reproduce in their prime. This fundamental principle underpins all three complementary frameworks we're about to explore, each offering a different lens on the same underlying phenomenon of why aging persists.
Okay, quick recap then before we dive into those three frameworks. So far, we've seen how a safer environment for those Sapelo Island opossums actually led to longer, healthier lives. And that's because natural selection effectively has a diminishing interest in keeping us perfectly maintained once we've passed on our genes. It's almost like a warranty running out after the main job is done.
Exactly. This foundational principle is what allows these three theories, mutation accumulation, antagonistic pleiotropy, and disposable soma to explain why aging is an evolved outcome, rather than just a simple, inevitable biological decay or a design flaw. It's a direct consequence of evolution's relentless focus on early life reproductive success. It's incredibly elegant when you consider it.
First up, let's dive into Sir Peter Medawar's groundbreaking idea, which he introduced back in 1952. This is where we get the concept of mutation accumulation.
Medawar's core concept for mutation accumulation is brilliantly simple. Late-acting deleterious mutations simply accumulate in the genome over evolutionary time. The underlying logic is directly tied to what we just discussed with Hamilton's declining force of selection. Selection against these mutations is incredibly weak because their negative effects only manifest after most reproduction has occurred. Think about it. If a mutation causes a problem when an organism is very old, say, past its reproductive years, then most individuals carrying that mutation have already passed on their genes to the next generation before the mutation ever becomes a problem for them. This means natural selection doesn't see or effectively weed out these mutations. They just sort of slip through the selective net.
So it's not that evolution wants these mutations, it's just that it can't effectively get rid of them if they only cause trouble late.
Precisely. Medawar's work was primarily a theoretical argument, a powerful piece of conceptual reasoning based on demographic logic rather than a specific lab experiment. The endpoint prediction of this theory is an age-increasing genetic load, meaning that over generations, more and more harmful mutations accumulate in the population that express themselves later in life, contributing to the diseases and frailties of old age. Medawar, of course, presented this as a theoretical framework, so there were no specific conflicts of interest or risks reported in the context of a study design. To use a classic analogy, imagine a car factory. They're obsessed with safety features and performance for the first few years of a car's life, right? That's when most people buy it. That's their reputation on the line. But if a specific part only fails after the car has been sold and driven for 20 years, well, the factory isn't under strong evolutionary pressure to fix that specific defect in its new models compared to a part that fails on day one. Natural selection operates with a similar, albeit brutal, logic.
Okay, that makes sense. So, bad stuff accumulates late because selection is looking the other way, focused on the early years. What's the next big idea?
Next, we have George C. Williams' framework from 1957, introducing antagonistic pleiotropy. This one really gets at the idea of trade-off.
Antagonistic pleiotropy. Okay, break that down. Pleiotropy means one gene does multiple things, right?
Exactly. Williams' core concept revolves around pleiotropy, a single gene having multiple, seemingly unrelated effects. Antagonistic pleiotropy specifically describes genes that confer significant benefit early in life, maybe boosting reproduction, accelerating growth, enhancing youthful vigor, but, crucially, have deleterious or harmful effects later in life.
So a double-edged sword, genetically speaking.
Precisely. The selection pressure here is profound. These genes are robustly selected for because their early life benefits are so strongly favored by natural selection. Those early advantages, which directly enhance an organism's ability to survive and reproduce, effectively override the weak selection against their late life costs. From an evolutionary perspective, it's a net positive gain in fitness, even with the downside later on. Williams's work, much like Medawar's, was a conceptual framework supported by various natural history exemplars he observed. The endpoint prediction of this theory is the existence of negative genetic correlations between early fitness traits, like fecundity or rapid growth rate, and late survival. Essentially, the genes that make you better at reproducing young also, in a way, make you worse at living long.
Can you give an example of that?
Sure. If we connect this to the bigger picture, it's like a biological Faustian bargain. A clear gain now for a debt that comes due much later. The trade-off is built into our very genetic architecture. For instance, imagine a gene that promotes rapid bone growth in adolescence. That helps build a strong, competitive individual, which is great for early survival and finding mates. But maybe that same rapid growth pathway contributes to bone density loss or increased fracture risk in very old age. The early advantage is so powerful that evolution effectively accepts the later cost.
That really highlights the trade-off idea. Okay, so we have mutations accumulating late, and we have genes that are good early but bad late. What's the third pillar?
The third is Thomas Kirkwood's disposable soma theory, which he first proposed in 1977 and elaborated on later. This theory brings an economic resource allocation perspective to the table. It really makes you think about the body as having a kind of budget.
Disposable soma. Sounds a bit grim, like the body's just a temporary vehicle.
Well, in a purely evolutionary sense, that's not far off. Kirkwood's core concept is that organisms face a fundamental, unavoidable resource allocation problem. They have limited energy and resources. They must budget these limited resources between two critically important needs. On one hand, you have germline transmission investing in reproduction, passing on your genes. On the other hand, there's somatic repair, maintaining the non-reproductive body, the soma, to ensure its survival and function long enough to reproduce.
Okay, so it's a choice. Reproduce or repair.
It's a balance driven by risk. The budgeting problem becomes strikingly clear when you factor in extrinsic mortality, the chance of dying from predators, accidents, disease. Because late survival contributes less to overall fitness, remember Hamilton's declining selection force, and the risk of dying from external causes anyway. The optimal evolutionary policy is to invest just enough in somatic maintenance, not perfection. Spending too much energy on perfect, continuous maintenance would be evolutionarily wasteful if the organism is likely to die from external hazards long before it can reap the benefits of that perfect repair.
So why build a body to last 100 years if you're likely to get eaten in 5?
Exactly. The body, from an evolutionary perspective, has to make a cold, hard calculation. Is it worth the investment to be perfectly maintained if I'm probably going to get eaten tomorrow anyway? Better to divert those resources to making sure you have offspring now. The endpoint prediction from the disposable soma theory is quite clear. A higher extrinsic hazard selects for lower investment in maintenance and, consequently, faster senescence or aging. It's essentially an economic model for the body. Why invest in perfect upkeep for a machine that's likely to crash soon anyway? Better to put resources into making more machines.
And these 3 ideas, mutation accumulation, antagonistic pleiotropy, disposable soma, they work together.
Yes, it's important to understand that these 3 frameworks are not competing theories. They are complementary, providing different but consistent lenses on the same phenomenon. And critically, they have significant empirical support. We've already discussed the Opossum Island mainland natural experiment, a beautiful real-world example. Beyond that, numerous comparative analyses across species bolster these theories. For example, species with lower extrinsic mortality, like birds and bats which can fly away from danger, tend to evolve significantly longer lifespans compared to similarly sized terrestrial mammals. Within species, predator release on islands often leads to longer life history traits, echoing Osted's opossum study. And Hamilton's mathematical models from 1966 rigorously established the age-specific selection gradients that provide the underlying quantitative basis for all these ideas. They all point in the same direction.
Okay, that's a lot of foundational evolutionary theory to unpack, but it's crucial for understanding aging. To put a pin in this, let's recap the 3 core evolutionary whys of aging, like a quick flashcard summary.
Absolutely. Think of these as the 3 pillars supporting our understanding of why aging exists. Here's your flashcard summary for this crucial section. First, mutation accumulation. Medawar. Deleterious genes whose effects only show up late in life aren't strongly selected against because they appear after reproduction, so they just build up in the population over time. Second, antagonistic pleiotropy. Williams. Genes that give big early life benefits, like boosting reproduction or growth, are strongly favored by evolution, even if they come with significant late life costs like disease or decline. That's a trade-off. Third, disposable soma. Kirkwood. Organisms have to budget their limited resources. Evolutionarily, it's often optimal to invest just enough in body maintenance and repair, not strive for perfection, especially if external dangers like predation are high. This prioritizes reproduction over longevity. And the connecting principle. All 3 of these arise fundamentally from the weakening force of selection with age, particularly after the peak reproductive period. Evolution's focus shifts away from maintaining the individual indefinitely.
Okay, so we've established the why from an evolutionary standpoint. Now, how do these grand theoretical ideas actually manifest? What are the precise biological mechanisms in our bodies that reflect these age-declining selection gradients and antagonistic trade-offs? This is where it gets really tangible.
It does. Let's start with the age-declining selection gradient itself. The strength of natural selection on survival at any given age, let's call it age X, is directly proportional to the expected future reproduction beyond that age. So if you're young and have a lot of reproductive potential ahead of you, selection is incredibly strong to keep you alive and healthy. However, as an organism ages, fewer individuals remain alive to reproduce, and consequently, the marginal fitness gain from any improvement in survival or reproduction at that older age shrinks significantly. It's just demographics.
Fewer potential offspring means less evolutionary incentive to keep you going perfectly.
Exactly. The direct consequence for genes is profound. Variants that are deleterious only after a certain age, say after age 60 in humans, face a much weaker purifying force of selection compared to variants that cause harm at a younger, reproductively vital age like age 20. This explains why alleles, or gene versions, with late pathogenic effects can become surprisingly common in a population. A plausible human example is the ApoE4 allele. This gene variant is strongly associated with an increased late-life Alzheimer's risk. Yet it may have persisted and even thrived because it possibly conferred early life advantages in certain ancestral environments, perhaps in lipid transport or maybe enhancing infection responses. The potential early benefit outweighs the later cost, evolutionarily speaking.
So that's mutation accumulation and maybe even antagonistic pleiotropy working together on one gene.
It can be seen that way, yes. This logic also helps explain why tumor suppressor pathways in our bodies show designs that often involve a trade-off. Effective youthful surveillance against cancer, a massive early life survival benefit, often comes at the cost of reduced later tissue maintenance and regenerative capacity, contributing to aging signs. The overall empirical signature isn't just a single gene. It's a consistent pattern of negative genetic covariance between early fertility traits, like reproductive output, and late survival. Longitudinal studies in wild populations have repeatedly documented such trade-offs.
Williams's hypothesis about antagonistic pleiotropy isn't just theory. It really gains teeth when we ground it in actual biological pathways. Can we look at some key examples that illustrate this antagonistic tug-of-war within our own systems?
Absolutely. The elegance of antagonistic pleiotropy is how it manifests at the molecular and physiological level. Let's look at a few key examples. Take the growth hormone IGF-1 signaling, GH-IGF-1 pathway. In early life, this pathway is a powerhouse. It strongly accelerates growth, promotes sexual maturation, and significantly increases fecundity or reproductive capacity. These are all clear fitness advantages, heavily favored by selection.
Sounds great early on. What's the catch?
The late life cost is stark. This pathway is consistently associated with shorter lifespan across diverse taxa. Experimental evidence in mice is compelling. Loss-of-function mutations in this axis lead to Ames dwarfs and smell dwarfs. These mice dramatically live longer and delay the onset of multiple age-related diseases. A specific study back in 1996 showed lifespan increases up to 40–60% in these mice, plus better glucose regulation and less oxidative stress. This is high-confidence data in mice, widely replicated.
So faster growth early means faster aging later in this case.
That's the trade-off it represents. You can imagine it like a growth accelerator pedal in a car. It gets you up to speed quickly, helps you win the reproduction race, but maybe it wears down the engine faster over the very long run.
Okay, what's another example?
Another canonical one is p53, often called the guardian of the genome. This gene is a perfect embodiment of this trade-off. Hyperactive p53 variants are powerful tumor suppressors. They prevent cancer, which is an enormous early life survival benefit, especially in rapidly growing organisms.
That sounds purely beneficial. Where's the antagonism?
The flip side is that these very same hyperactive p53 variants also induce early senescence phenotypes, essentially causing cells to age and stop dividing prematurely, and they impair tissue renewal. This contributes to the signs of aging we observe. Mouse models consistently demonstrate this. Mice engineered with hyperactive p53 show fewer tumors but paradoxically exhibit accelerated aging signs. It's a high-confidence finding, intrinsic to the mechanism, and widely replicated. Powerful cancer protection early comes at the cost of faster tissue aging later.
Makes sense. Cancer is often a disease of aging, but the protection mechanism itself contributes to aging. What about hormones?
Right. Let's look at human testosterone, a classic example particularly relevant for males. Higher early testosterone levels correlate with crucial mating success, greater muscle accrual, competitive advantages, all driving reproductive fitness.
The evolutionary upside is clear.
Yes, but these same elevated levels are linked to increased risks later in life, such as prostate growth and potential adverse cardiovascular signals. Evidence comes from human cohort associations, tracking things like heart disease and cancer proxies. While effect sizes can vary and replications are sometimes mixed, the overarching theme is undeniable. The architecture of our biology often means that what powerfully helps reproduction and growth early frequently taxes long-term maintenance. It's a fundamental design principle, not an accident.
Okay, so we've seen how selection's grip weakens with age, allowing harmful late-life mutations to persist, and how antagonistic pleiotropy forces our bodies to make that deal. Quick gains now for potential costs later. The GH-IGF-1 axis, p53, and even testosterone are perfect, mechanistic examples of these trade-offs in action, proving these aren't just abstract ideas. It makes a lot of sense, actually, once you lay it out that way.
It does. These aren't just theoretical constructs. These are fundamental biological pathways, illustrating that aging isn't a design flaw or simple decay. It is, in fact, an evolved outcome of prioritizing early life fitness and reproductive success above everything else. Our bodies are incredibly efficient at this prioritization.
Now let's revisit Kirkwood's disposable soma model from the perspective of cellular budgeting and error tolerance. How does this economic view play out at the cellular and molecular level? It feels like we have a biological accountant somewhere in our cells managing resources.
That's a great way to put it. Kirkwood's model explicitly treats crucial maintenance processes, things like DNA repair, proteostasis, which is protein quality control, making sure proteins fold right and clearing out bad ones, and other cellular repair mechanisms as biologically costly. They require energy and resources.
Resources that could be used for something else, like making offspring.
Exactly. The good enough principle is central here. When extrinsic mortality, the chance of dying from external causes, is high, spending valuable, limited resources to achieve near-perfect cellular repair yields very little fitness return. It's simply not worth the investment from an evolutionary perspective. The optimal strategy is to invest good enough resources, leading to residual damage accumulating over time, which then expresses itself as aging.
So the body doesn't bother with perfect upkeep if it thinks it won't last long anyway.
That's the core logic. This model also rationalizes why many maintenance pathways aren't constitutively maximal, meaning they're not always running at full throttle. Instead, they're often inducible and stress responsive. They get activated when needed, suggesting a controlled, economical allocation rather than continuous overinvestment. Furthermore, disposable soma helps rationalize why caloric restriction, or CR, and slowed growth, particularly when external danger is low, can extend lifespan. By reducing resources devoted to rapid reproduction and growth, more can potentially be diverted to cellular repair and maintenance, effectively shifting the internal budget.
This budgeting idea leads us directly to one of the most exciting and actively researched areas of aging science today: nutrient sensing. This isn't just random biochemistry, is it? It really sounds like an intricate, evolved system for resource management, a master controller.
Absolutely not random. It's a beautifully evolved, sophisticated system. It acts as an integrated control panel for resource allocation within the cell and the entire organism. This system comprises 4 key signaling pathways that you often hear about: the insulin IGF-1 signaling ISEF pathway, mTOR, mechanistic target of rapamycin, AMPK, AMP-activated protein kinase, and sirtuins.
Okay, those are the big players in aging research right now. How do they act as controllers?
Together, this system constantly senses the abundance of nutrients in the environment and the metabolic state of the cell. It functions like a finely tuned toggle switch for the body's entire resource allocation strategy. When there's a plenty, signal high nutrients, you see high activity in ISIS and mTOR, and low activity in AMPK. This state prioritizes energy allocation towards things like protein synthesis, cell growth, cell division, and reproduction. The body is effectively in full-on growth and reproduce mode.
Got it. Growth mode when food is abundant. What about when it's scarce?
Conversely, when there's a scarcity signal, low nutrients, or maybe environmental stress, you see low ISMTOR activity combined with high AMPK and sirtuin activity. This shifts the priority dramatically towards maintenance and recycling. It upregulates crucial processes like autophagy. The cell's self-cleaning mechanism enhances DNA repair and generally boosts overall stress resistance. The body switches into repair and survive mode, conserving resources and shoring up defenses.
So it's an evolved switch that dictates grow now or survive longer.
Precisely. This isn't just a random set of biochemical reactions. It is a sophisticated, highly evolved allocation policy, fine-tuned over millennia. And the evidence is strikingly consistent across diverse organisms. From simple yeast and worms like C. elegans to fruit flies and mice, consistently downshifting ISMTOR activity or upshifting AMPK sirtuin activity lengthens lifespan and delays the onset of age-related diseases. This consistency across vast evolutionary distances strongly supports the disposable soma theory, demonstrating a conserved biological strategy for managing aging in response to resource availability. It's truly elegant.
Wow, we've gone from theoretical principles to the very molecular switches that dictate our aging. It's like we just opened up the hood of the evolutionary engine and can see the levers. Let's recap this crucial section on mechanisms.
Definitely. This section really shows us how these grand evolutionary theories are grounded in biological reality, providing mechanistic explanations for what we observe. So if you're looking for the essential points from this section, here they are. First, that age-declining selection isn't just theory. It means selection against harmful genes weakens after reproduction, letting late-acting problems, like those linked to ApoE, APO4, persist. Evolution just doesn't prioritize fixing late-life issues. Second, we saw mechanistic antagonistic pleiotropy in action. Genes providing early benefits like GHI-GF1 for growth, P53 for tumor suppression often carry late-life costs. These biological Faustian bargains are built into our design. Third, disposable soma in practice explains our body's resource budgeting. Maintenance is costly, so our systems invest just enough in repair, especially if external risks are high, prioritizing reproduction over indefinite longevity. And finally, we discuss nutrient sensing pathways, isomtor, APK, and sirtuins. These aren't just random reactions, they are evolved allocation controllers. They act as a toggle switch, shifting the body between growth reproduction, high nutrients, and maintenance air repair, low nutrient stress. This is how the body manages its budget in real time.
With this deep understanding of evolutionary trade-offs, the ancient give and take that shaped our aging, we can now really appreciate how various modern interventions try to cheat these ancient, evolved biological policies. It's not about magic. It's about intelligently shifting that biological budget.
Precisely. Let's start with caloric restriction, or CR. Its mechanism aligns perfectly with the disposable soma and nutrient sensing ideas. CR acts by simulating those scarcity signals we just discussed. It prompts the body to reallocate resources away from rapid growth and reproduction and towards maintenance and repair. In rodents, the evidence is incredibly robust. Lifelong 20-40% CR consistently extends both lifespan and health span. High confidence here.
Okay, works well in rodents. What about closer to humans, like monkeys?
Right. When we move to rhesus monkeys, the picture gets nuanced, showing the complexities. The University of Wisconsin study reported significant survival benefits and delayed disease in their CR group. Very encouraging. However, another major study from the NIH-NIA reported improved health markers, but without a clear lifespan gain versus their well-fed controls.
Why the difference?
A key factor seemed to be the control diet itself. The NIA monkeys were already on a healthier baseline diet. A later joint analysis concluded that CR benefits depend heavily on diet composition, age at initiation, and baseline body fat. So context matters hugely. It's not a simple universal effect.
And in humans.
Yeah.
I can't really do lifelong CR studies for a lifespan.
Exactly. For human data, we look at shorter trials and biomarkers. The Calorie Phase 2 trial involved about 218 non-obese adults undergoing 25% CR for 2 years. They showed significant improvements in cardiometabolic risk factors, high confidence data replicated. More recently, a follow-up showed a modest but significant 2-3% slowing of biological aging using DNA methylation clocks.
So, signs of slower aging, but not proof of longer life yet.
Correct. It suggests an impact on the rate of aging, which is exciting, but event-level outcomes, actual longer lifespan or reduced disease incidence remain untested in humans for CR. It requires decades-long trials, which are hard to conduct.
Okay, what about drugs that mimic CR, like rapamycin?
Yes. Rapamycin is a powerful cheat because it directly inhibits mTORC1, a key part of that MTR nutrient sensing pathway. By doing so, it imposes a maintenance-favoring state, mimicking aspects of scarcity. The mouse evidence is compelling. A landmark 2009 study used genetically diverse mice and, crucially, started treatment quite late, around 600 days of age, equivalent to maybe a 50 or 60-year-old human. Despite this late start, rapamycin significantly extended median lifespan by 9% in males, 14% in females, and increased maximum lifespan too. High confidence in mice, widely replicated, though there's a moderate risk of glucose intolerance noted. Later studies confirm these benefits, even with intermittent dosing to reduce side effects.
That late start effect is really interesting. What about human data for rapamycin?
For human data on rapamycin or its analogs, or rapalogs, we look at functional outcomes. Trials in older adults showed that low-dose mTORC1 inhibition improved vaccine response about 20% higher antibody titers and reduced reported respiratory infections, promising for immune function. However, confidence is moderate. There are risks like mouth sores, stomatitis, and potential wound healing issues. And importantly, replications have been partial. There was a significant null result. A large phase 3 trial testing a rapalog, RTB101, failed to meet its primary endpoint of reducing respiratory infections. So while biomarkers might improve, clinical event-level benefits in humans aren't guaranteed and need rigorous validation.
Yeah, this is where it gets particularly interesting, highlighting how deep these evolutionary trade-offs run, even manifesting differently between sexes. Can you tell us about acarbose and 17E estradiol? Seems like they're uncovering some subtle yet profound differences.
They absolutely are. These two interventions, tested extensively in the NIH's Interventions Testing Program, ITP, illustrate fascinating underlying sex-specific trade-offs. Acarbose, which is the drug that blunts blood sugar spikes after meals, extended male mouse lifespan by roughly 22%. That's a substantial gain. However, in females, the effects were much smaller or inconsistent, sometimes non-existent.
Wow, a huge difference. What about the other one?
17-estradiol. This is a non-feminizing estrogenic compound, meaning it has some estrogen-like effects without causing female characteristics. It extended male mouse lifespan by about 12-19%, again, a solid effect. But similar to acarbose, no significant benefit was observed in females.
So why this stark difference between males and females for these specific drugs?
These sex dimorphisms likely reflect the different sex-specific endocrine baselines, the different hormonal environments males and females operate under. They also reflect the differential costs and benefits of modulating pathways like insulin IGF-1 signaling, which acarbose affects indirectly, and inflammation between the sexes. Evolution has sculpted distinct resource allocation strategies based on the different demands of reproduction for males and females. These interventions seem to tap into those deeply embedded differences. What's compelling is the consistency. In these well-conducted mouse studies, both drugs gave a solid 12-22% lifespan boost, but almost exclusively in males. High confidence, replicated finding, highlighting these crucial sex differences we must consider for human translation.
One of the most encouraging findings from all this research, for me, is that our biology isn't fixed, especially in later life. There's real late-life plasticity. It's not a one-way street of inevitable decline. That really offers a lot of hope, doesn't it?
It's a critical point, and it's powerfully demonstrated by that 2009 rapamycin study we mentioned. The fact that rapamycin could extend lifespan in mice even when treatment started late, around 600 days, equivalent to human middle to late adulthood, directly undermines the claim that aging is irreversibly fixed. It shows that the underlying biological pathways, the maintenance programs, remain inducible and responsive even later in life. This is incredibly significant for future human interventions. It offers hope that we don't necessarily have to start treatments from birth to see benefits. The body retains the capacity to respond.
So we've explored how caloric restriction, rapamycin, and even those sex-specific compounds like acarbose and 17-estradiol are all attempts to cheat our evolved aging pathways with varying success and specific trade-offs, so that we're trying to reprogram our ancient operating system.
And critically, these studies highlight that the body retains significant plasticity even into late life. This is a powerful message for therapeutic development. It suggests that targeted interventions can indeed influence the trajectory of aging, rather than just being futile attempts against an immutable process. It gives us a real scientific basis for optimism, but also for caution.
Looking beyond humans, the animal kingdom offers incredible insights into the design space of longevity. It shows what's truly possible, the outer limits of how long a mammal can live, and how different evolutionary pressures lead to vastly different outcomes.
Indeed. These comparative biological studies are crucial. Take naked mole rats. These fascinating creatures show negligible senescence by some metrics. Their risk of dying barely increases with age across much of their lifespan, defying the usual Gompertzian aging pattern. A 2018 study confirmed this near-flat hazard function up to advanced ages. High confidence here. Their longevity is linked to extraordinary proteostasis protein quality control, exceptional DNA repair, and an unusual extracellular matrix component, high molecular weight hyaluronan, implicated in their remarkable cancer resistance.
They sound almost alien. What about something bigger?
Then there are bowhead whales. They can exceed 200 years of age, the longest living mammals. Genomic studies revealed specific adaptations in their DNA repair genes, cell cycle control, and insulin signaling pathways. Clear signs that selection heavily favored robust maintenance mechanisms in these giants.
And you mentioned bats and birds earlier.
Right. Bats and birds routinely outlive terrestrial mammals of similar mass, sometimes by decades. This is strongly linked to their lower extrinsic mortality. Flight reduces predation risk significantly. This observation strongly supports Hamilton's predictions. When environmental hazards are lifted, selection can indeed ratchet maintenance up.
So what's the lesson from these extreme examples?
The lesson is that mammalian biology can support far slower senescence than humans currently exhibit. Our lifespan isn't necessarily the ultimate limit. However, these evolved solutions often involve highly unusual cancer suppression tricks or specialized metabolisms. They're hard to just retrofit into human biology without potentially significant costs or risks. They offer a design blueprint, maybe inspiration, but not a simple copy-paste solution.
This brings us to a critical, unavoidable frontier, perhaps the most profound trade-off of all: the inherent tension between pushing for regeneration and the inherent risk of cancer. It seems our bodies have already made a choice here.
That's right. It's a fundamental biological conflict. Tumor suppression and regenerative capacity are, in many ways, antagonistic objectives. Enhancing cellular proliferation, making cells divide more, or resetting epigenetic controls to rejuvenate tissues, while potentially restorative, inevitably carries the significant risk of oncogenesis, cancer development. The very mechanisms that help us grow and repair can, if unchecked, lead to dangerous, uncontrolled growth.
And specific pathways embody this conflict.
Key pathways like P16INK4A and the P53 network act as critical brakes. They are powerful tumor suppressors, a huge early life benefit, but in doing so, they also promote cellular senescence, cell aging, and reduce the regenerative potential of tissues over time. It's a trade-off embedded right into our cellular defense mechanisms.
We see this playing out in new experimental therapies.
Yes, consider partial cellular reprogramming. This involves briefly expressing the OSK Yamanaka factors to make cells more youthful. In mouse models, it can rejuvenate some tissues, like restoring function in optic nerve regeneration models. Remarkable stuff. However, the trade-off is stark. If these factors are overdriven or mistargeted, they can cause teratoma's bizarre tumors and even kill the animal. So while promising, the risk is high if dosing isn't perfectly managed. Moderate confidence, high risk if mismanaged.
What about fixing telomeres? That sounds less risky.
Telomerase activation aims to restore the replicative capacity of cells by maintaining telomeres, the protective caps on chromosomes. This could counteract cellular aging. However, this inherently carries the potential risk of raising cancer risk due to allowing uncontrolled cell division. In mouse models, gene therapy to activate telomerase did extend median lifespan by about 24% without obvious cancer increases, but this was under carefully controlled dosing. The risk is context-dependent. While some replications exist, there's potential for tumor promotion if not managed precisely. Again, a razor's edge, moderate confidence, context-dependent risk.
So how did things like elephants solve this? They're huge and live long, but don't get proportionally more cancer.
That's Peter's paradox. The solution wasn't an absence of trade-offs, but the evolution of extra tumor suppressor gene copies and sophisticated circuit redesigns. Elephants, for instance, have multiple extra copies of the TP53 gene, giving them heightened cancer resistance. The evolutionary tension is explicit. Selection in long-lived lineages favored robust tumor suppression, even at the cost of potentially slower repair and slower aging. Any cure for aging must fundamentally respect and navigate this cancer constraint. It demands incredibly clever safeguards, tight spatial and temporal control, built-in suicide switches for rogue cells, vigilant surveillance. Evolution made a choice.
This all comes back to the idea of biological budgeting, which is perfectly encapsulated by the nutrient-sensing pathways we discussed earlier. They really are the central control panel for these evolutionary trade-offs, aren't they?
Yeah.
It feels like every intervention is just trying to hack this one core system.
They absolutely are. The allocation controller triad, that interplay between mTORus versus AMPK sirtuins, directly implements the soma's budget rules that Kirkwood theorized. When nutrients and growth cues are high ISMTOR, organisms are programmed to spend on reproduction and growth, actively suppressing maintenance like autophagy. When nutrient cues are low or stress is high AMPK sirtuins, the body defers immediate reproduction and robustly upregulates maintenance and repair pathways.
And the interventions are just levers on this system.
Exactly. Caloric restriction does it behaviorally. Rapamycin does it pharmacologically by hitting mTORC1. A carbose dampens glucose spikes and downstream acetone. 17-acetradiol modulates nutrient signaling in specific tissues with those sex-dependent effects. The cheat sheet logic for future interventions is find ways to keep maintenance pathways high without catastrophic fertility costs or impairing essential functions like wound healing. If you can do that, you might push the organism into slower aging.
But there's always a catch, a guardrail.
Always. Every push has a cost that must be managed. Repalogs risk immunosuppression and stomatitis. CR risks muscle and bone loss, especially in the elderly. Poorly dosed mTOR inhibition risks glucose intolerance. And we saw the sex-specific trade-offs. Translating these to humans means proactively defining adverse event sentinels, critical safety readouts that must be tracked alongside any aging surrogate. Things like impaired wound healing, infection rates, anemia, bone density, muscle strength. These aren't just side effects. They are potential costs of cheating evolution, and safety must be paramount.
Wow. We've seen how interventions are precisely targeting these evolutionary trade-offs and how animal models like the naked mole rat show us the incredible plasticity possible. But this also means confronting the uncomfortable truth that pushing regeneration often comes with the inherent unavoidable risk of cancer. It's a tightrope walk.
It truly is, and that final point is key. Evolution has made a profound, ancient choice to accept aging as a consequence of prioritizing early life fitness rather than widespread, ubiquitous cancer. This fundamental trade-off is deeply written into our circuits. Any strategy to extend healthy human lifespan must operate within these constraints.
No scientific story is ever perfectly clean. Our source material makes sure to include important nulls and counter-evidence, which refine our understanding, rather than outright refuting the core theories. These complexities are essential for a nuanced perspective, aren't they?
Indeed. These disciplined nulls are just as informative as the successes, sometimes more so. For instance, the mixed survival results for caloric restriction in rhesus monkeys across those two big studies, Wisconsin and NIA. They don't invalidate CR's potential, but they profoundly emphasize that its success isn't absolute. It depends heavily on diet quality, baseline health, age of starting. A vital warning against naive enthusiasm.
And that rapamycin trial failure.
All right, Restorbio's RTB101 phase 3 failure. This human trial used an mTOR inhibitor aiming to reduce respiratory infections in older adults. It failed to meet its primary endpoint, even though earlier studies show improved immune biomarkers. It clearly reminds us that modulating mTOR might improve markers, but it's not a guaranteed event-level win in humans. Clinical benefits need rigorous proof in large trials. High confidence in this null result.
What about senolytics, clearing old cells?
Another example, Unity Biotechnology's UBX0101 trial for knee osteoarthritis, a senolytic approach, missed its pain and function endpoints. This serves as another trade-off caution for interventions targeting complex late-life pathologies. These nulls don't falsify the core evolutionary frameworks. Instead, they serve as crucial warnings. Cheating evolved trade-offs is often significantly harder and more nuanced in practice than it appears in theory or simpler models.
And it seems like these nulls also highlight the importance of sex dimorphism. Williams's theory would predict sex-specific outcomes, wouldn't it?
Absolutely. Williams's theory of antagonistic pleiotropy, focusing on early reproductive benefits versus late cost, fundamentally predicts sex-specific optima. Why? Because males and females have different reproductive schedules, metabolic demands, hormonal environments. The observed male bias benefits with a carbose and 17E2 in mice are textbook confirmations. These differences aren't bugs. They're powerful signals that the levers we pull intersect with deeply evolved, sex-specific strategies. The implication for human translation is critical. Trials must anticipate and design for sex interactions a priori, from the start, not as an afterthought. We can't assume interventions work the same way in everyone.
Let's reiterate a crucial point about late-life plasticity. The idea that our biology isn't fixed, even as we get older, seems profoundly hopeful.
It is. That 2009 rapamycin study showing lifespan gains, even when started late in mice, profoundly implies that fundamental maintenance programs remain inducible. The biology isn't irreversibly fixed. This means we don't necessarily have to start treatments from birth, offering significant hope for therapeutics in an aging human population.
But there are limits, presumably.
Of course, this plasticity isn't uniform across all tissues or conditions, and it certainly has limits. Some tissues might be too degraded. The optimal dose window might narrow significantly with age, making treatment trickier. In humans, we see this plasticity clearly with lifestyle. Starting exercise even in your 70s brings meaningful gains in aerobic capacity, gait speed, strength, high confidence, low risk, widely replicated. Pharmacological plasticity, how much drugs can help late in life, is less mapped out in humans. That's a key agenda for future clinical trials.
Finally, let's reflect on comparative biology as a design space, not destiny. The naked mole rat and bowhead whale are compelling proof that mammalian biochemistry can support far slower senescence and vastly extended lifespans than humans typically experience. It really does make you wonder why we don't live that long.
It proves our current lifespan isn't some immutable biological ceiling. However, it's critical to remember these solutions evolved under very specific ecologies: burrowing life for mole rats, deep-sea life for bowheads. These weren't minor tweaks, but involve system-wide biological rewiring affecting metabolism, cancer suppression, everything. Profound adaptations.
So they can't just copy them directly?
Yeah. No. The actionable lesson isn't to become a mole rat. It's to carefully identify which specific maintenance modules, maybe enhance DNA repair, better proteostasis, are potentially portable into human therapeutics without causing intolerable trade-offs or systemic instability. It's about smart engineering inspired by nature, not mimicry. This brings us back one last time to that most unavoidable frontier: regeneration versus cancer. Peto's paradox: large, long-lived animals not riddled with cancer was solved not by avoiding trade-offs, but by evolving better solutions, like extra tumor suppressor gene copies, elephants and TP53. In humans, any attempt to push regeneration, like partial cellular reprogramming, will inherently require equally clever and rigorous safeguards. Tight control, suicide switches, vigilant surveillance. Evolution's profound choice was to accept aging as a consequence rather than widespread cancer. This choice is deeply written into our circuits. Any successful aging intervention must fundamentally respect this existential constraint.
This final section really grounds the evolutionary theory in the complexities of real-world biology and intervention. It makes you realize how many layers there are. So what are the crucial considerations for navigating the future of aging research? Like a final flashcard.
Exactly. These are the essential caveats and insights. Your final flashcard for navigating the complexities. First, null results are crucial. Mixed results, like rhesus-CR, RTB101, don't invalidate theory but show cheating trade-offs as complex and context-dependent in practice. Humility is needed. Second, sex dimorphism is predicted and observed. Evolution predicts sex-specific optima. Interventions like acarbose and 17E2 confirm this with male-biased effects in mice, reflecting evolved resource allocation differences we must account for. Third, late-life plasticity is real but limited. Programs remain inducible, for G. rapamycin late start, offering home for interventions even in older age. But limits exist, and pharmacological plasticity in late-life humans needs more study. Fourth, comparative biology maps design space. Animals like mole rats and whales show slower senescence as possible, but require depth systemic changes. Identifying potentially portable maintenance modules is the challenge, not simple mimicry. And finally, regeneration versus cancer, the unavoidable frontier. Pushing tissue rejuvenation inevitably risks cancer. Any aging intervention must include robust oncological safeguards, respecting evolution's profound trade-off.
So what does this all mean for you? We began this deep dive with the simple yet profound story of the Sapelo Island opossums. Their safer world led to slower aging, and that perfectly embodies the logic of selection gradients we've explored. It highlights that aging isn't a random breakdown, an accidental flaw, but rather a deeply evolved outcome, a strategic, if sometimes brutal, consequence of natural selection prioritizing early life reproductive fitness. The beauty here is that Medawar's mutation accumulation, Williams's antagonistic pleiotropy, and Kirkwood's disposable soma aren't competing theories. They are complementary explanations that converge on why aging evolves. And crucially, understanding these deep evolutionary roots helps us grasp why the most promising levers to slow aging often come with those edge costs or trade-offs that we must predict, manage, and meticulously account for. The goal isn't to negate evolution, but to intelligently navigate its constraints. The whole point of this deep dive was to replace the mystique around aging with concrete biological mechanisms, grounded in rigorous evolutionary theory, and to prepare you for the next steps in understanding how we might, with great care and intelligence, navigate these ancient, elegant trade-offs.
This raises an important question for the future. Considering these evolutionary trade-offs and complexities, what would truly change our collective mind about the malleability of human aging? What's the key evidence we're looking for? For me, it would be unequivocal human evidence that a carefully dosed allocation, cheap, perhaps intermittent MTO inhibition combined synergistically with lifestyle changes like targeted exercise and ensuring protein sufficiency can robustly reduce the time to multimorbidity. That means delaying the onset of multiple chronic diseases without materially increasing infections or impairing wound healing over, say, a solid 3 to 5 year trial period. That would be a game changer. Or alternatively, a well-powered primate study showing that sex and age-aware nutrient-sensing modulation extends both lifespan and, critically, disability-free survival. That kind of evidence would truly justify aggressive translational trials in humans. Just a quick but critical note, of course. While we've discussed these fascinating interventions in the science, this deep dive is strictly for informational purposes. Discussion is absolutely not a recommendation for self-experimentation. Off-label use of geroprotectors belongs only in ethically approved clinical trials or under competent, supervised clinical care with appropriate monitoring. Please be safe. This deep dive has laid the theoretical groundwork, clarifying that aging isn't a random breakdown, but an evolved conditional state shaped by the relentless logic of natural selection. Now, the question becomes not just why we age, but how precisely we might manage these ancient, elegant trade-offs in our quest for a longer, healthier life.